Originally, I come from the field, which is now called organ on chip, but it started from the MEMS, so the actuators and sensors. And we miniaturize the body. So we create the body on chip and organ on chip, and now it's a field of research which is presented in European community as well. We have European Organ on Chip Society already established since 2019. And now we have about 1,000 members, so we are growing field. And coming from the field of micro and nano electronics, where everything is organized and beautiful, and you have the crystal grid of the materials. During my PhD, I came to this, and this is our immune system. It's a messy. It's what is going on here directly. The red, they are the phagocytes, and they are hunting. So this is like a computer game. And this is the video which is recorded over 1 hour. And it's speeded up. So that's exactly what's going on in your body right now. They are doing it every second of our life. They are moving, they are sensing, they are living, they produce energy, consume food and nutrition. They do a lot of things, and also they adapt. And this is the worst case of why we are here, and we will be many, many years in this field more, because those creatures adapt. It influenced my research a lot. The organ on chip I have done together with my colleague from Thailand. We were working at University Hospital in Magdeburg. And we have studied the stomach on chip. The reason was that we want to miniaturize the stomach as a model. So we have built the microfluidic device with the sensors inside. And we have cultured there the patient cells, so the primary cells from the patients, which have produced the mucous layer, which we have in the nose, for example, those saliva. This is important. And there, bacteria, they live. And the research groups from Italy of Baccelato, they have done really cool experiments in the Petri dish, where bacteria lives in those mucus, and it over accumulates. So every week bacteria has chance to survive because there is no flow. But in our stomach, we eat something, hopefully good things, or we drink cola, and on top of the bacteria, cola is flushed. So bacteria are dead if they were weak and they could not attach to the stomach epithelial layer. So that's what we have recreated and published together with Tonnam. And the interesting thing, we integrated there also the immunocells, and they were also moving in those epithelial tissues. But we have published the research on the cell lines, on the cells which are adapted, so genetically modified to grow in a Petri dish. And on the patient cells, which are real, taken from the patient body, we are still under revision. Already, how many years I'm postdoc? About 3 years. So 3 years, our paper is still under revision. A success story, I understand, but the problem, what happened by us? The reviewers, they have asked to repeat the experiment. Dream on. First, our cells, they have mutated. So they have adapted to the environment. Okay. Then we took another patient cells, and we hoped the bacteria will be the same. And we took Helicobacter pylori. No, bacteria has adapted as well. And then we cannot repeat the same experiment because cells are different, but bacteria is not same anymore. they are reacting on the messages differently. Okay, we are still under revision. We don't give up. We still repeat this experiment over and over. And we hope after some certain time, reviewer will be satisfied and will allow us to publish now a lot of the patient data. So this paper gained better and better. And that's how I started to work on the a bit organization of this field. And every crosstalk or every talk in between the cells, this is an example of the brain cells. Every talk in the biology looks like this. Every path which you see here, just as an example, includes much more factors and much more communication and crosstalks. And the molecular communication field, I got so great introductions now already. This field is emerging right now, and I want just to add why it emerges. It's why it's so popular now and gain more and more popularity. Because first of all, we know already a lot, and we need somehow to structure our knowledge. And the second, which is the very promising part for the future or for over 10 years future, is this immunotherapies and the mitochondrial therapies. I am not sure, have you ever heard about the mitochondria? Probably half of you. So mitochondria, it's the organelle inside of the cell. So we go deeper and deeper and we study deeper and deeper. And mitochondria, previously we thought that it produces the ATP molecules, which are energy molecules for our body. Now we know it's not only producing ATP molecules, it's also decision maker. So decision making is important. And mitochondria is involved in the crisis, or if you have lack of energy, okay, you forgot to take breakfast today. So, okay, you don't have nutritions which you need right now at the moment, and some of your colleagues take out the gun and say, okay, let's talk now, and you have stress and you need to run, for example. And at the same time, at the desk, you had some virus. For example, corona, somebody left. And you have the virus attack at the same time. What your body will do? Protect from the virus or run away from the neighbor, which has gun. So, and for this, mitochondria will take place in the decision. I hope we will run first, but let's see, maybe virus is more dangerous. So in this case, complexity is growing, decision-making is important, and those treatments and therapies, which are immuno-based and mitochondrial-based, they are totally based on the molecular communication between those cells and between all cells in the organism. So, variety of papers were published, too much. I cannot read all of them. It's over thousands of the papers about those therapies. Immunotherapy is already in the hospital available. There are still problems to de-weaponize those immune cells, but they will do. And mitochondrial therapies, they are arise right now, and they start from the depression treatment, because that's already one thing what is proved. Mitochondria and depression, they are connected, highly connected. And as a case study, that's what we do usually in the organ on chip field. We take the case study or we take the disease and then we have a look inside. We collect the knowledge which we have already, and then we can recreate this model physiologically on the miniaturized scale to study it. Recently, I watched a movie about the Stephen Hawking. I could recommend. It's a nice movie, so you can. And I took his case because it's very interesting. Why it's interesting? Because he's unique, or his case is unique. Because usually with this disease, you survive 5 years, not more. He survived 50. And this man had divorced twice, had 3 children, even in such a bad disease condition. So life is progressing always. This is the amyotrophic lateral sclerosis, which, if we discuss engineer-friendly, because I'm an engineer, I need those engineer-friendly information, very concrete. If we say engineer-friendly, then the system, the immune system, which I showed you on the first slide, which is moving, this system from protection turns into the destroy mode. And this destroy mode, for sure, for us, it's not beneficial. The way how it turns, it's very complex. So first layer, we can peel it as an onion, layer by layer. First layer, we get the genetic reason for this. But about 10% of the cases, we know it's genetic. 90% of the cases, they are sporadic, we don't know. Or we cannot rely that they are genetic. Here we can point those 90%. If the genetic information there plays important role, then it's those jumping genes, which are expressed because the environmental influence on the body. So then second layer will be the protein misfolding. When you have already not expressed in the correct way genes and the system starts to break, then the proteins, they accumulate inside of the cells, which is damaging. Cells, they understand it, they feel it, they sense signals. Also, it leads to the mitochondrial dysfunction, oxidative stress, a lot of processes which run not correct. And the system is still okay, but already under the stress. Then the layer 3 happens, immune system comes to play. Immune system comes there and try to fix the situation. And by fixing the situation, it comes into the destroy mode. And in this case, we start to have the degradation, exactly those motor neurons degradation. The layer 4, it's when the system comes into the feedback loop. So we have more degradation, so means more debris, more proteins which are leftovers, and we have more immune cells which come to help and rescue, and they do more damage, and so on. So we turn into the feedback loop, which if we try to reproduce as a theory, or if we would like to analyze it, we could take even the simplest model. I know in communication theory, you can introduce very complex models. There are, but I would not have so much time to stand here today and to present more complex models. So I took the simplest one, also good for me, because I'm the hardware person. So with the theories, I use them, but I don't like them so much. But here, the beauty of this, with this theory, we can reproduce those 3 main steps of the damage in this use case. First, what happens? Inflammation destroys motor neurons. For this, we have the equation. Neuron damage produces debris. So this we can also represent with the factors. And debris activate the inflammation through the nonlinear switch. And for this, we can also deploy the coefficients. The interesting part, or why we would like to talk about the Stephen Hawking, those parameters, quality, which are pointed there. So the neuron sensitivity to inflammation, immune amplification gain, inflammatory damping, and damage to debris conversion rate. Those parameters, they are unique to every person. They are not same from me and you. That's why the disease would also go into the different way or at different speed, because all those equations, they are time dependent. If we talk about those parameters and we go into the, thanks. And we talk about the Stephen Hawking, or what was different in his case. He was able to stay all the time near the threshold of his disease. And this is extremely hard, 50 years. He did not move to the collapsed phase, and he did not move to the healthy phase. He was always in this green region, dancing around the threshold, which is the most critical part for any disease. And then if you below threshold, then, or slightly near the threshold, in this use case, you have slow degeneration. That's why for him, it represents in the way that the typical sclerosis in this case would have the amplification gain high. By him, it was moderate. Damping, usually low, but by him, it was high. So all the parameters, they were different from the normal disease lasting. Then he had also the sensitivity to the toxicity lower than normal. So in this case, we could talk about the survival expectations. This is a parameter which usually involves much more factors. And in simple theories, we should not talk about this, because this is a topic which deserves to involve a lot of things into these thoughts. But at least to point out, for the most important thing, what gained us the knowledge of the disease of Stephen Hawking, also about our nowadays knowledge, that the small things, they matter. So it means dancing around the threshold about the disease, even the change of 10% in one of those parameters, it can change the survival rate a lot. And there are parameters which influence linearly and non-linearly. And for every disease, there are parameters which are more important and less important. And by modeling those diseases, we can make those expectations to which parameters we need to look more or pay attention more, which can cause us coming from the threshold or dancing around the threshold to the critical stage. Here, as you can see, the factor is not so much different, but it gives from 5 years to 50 years. And 50 years, I hope I will live maybe even without disease. So in this case, there are more factors, like in this case, for example, the damage to inflammation activation function. Those things we gain from the doctors and then from the hospitals which we work with. And I think the other groups, they also collaborate with doctors. By analyzing those data, we can address some more functions introduced to the theories, and therefore express the parameters better. In this case, just moving from the just involving one factor, we can here represent how the disease would last. And according to this activation function and damage level, we can plot the different graphs. They would be more correct and more corresponding to the real data if we address there more parameters for sure. But even with small changes in this nonlinear switch, the collapse of the disease, so going from the threshold to the extreme severe cases, it can be fastened to the 35%. So in this case, I want to point on the take-home messages. First of all, treating those sclerotic diseases or neurodegenerative diseases at all, it means restoring stability and not just killing inflammation, because inflammation was only in the beginning. And then this unstable system has to be tuned again. The difference between 5 years survival and 50 years survival can be pointed even with the simplest model. And the parameters can be given which influence the most the survival rate. Then a small molecular disturbance becomes a system-wide failure because damage and inflammation amplify each other. That is good to remember that our body is one system, and by introducing changes in one part, we can introduce the changes in other part of the body. And this is a closed loop system. There is always feedback. And therefore, mathematical models, they don't just describe disease, but also they reveal where intervention can shift the entire trajectory of the disease lasting, and therefore also the survival rate in the disease. So this would be highly important for the future. Thank you very much for your attention.
Thank you for the nice talk. So any questions?
Thank you. This is fascinating. So would your research, or in general your field and the direction you're taking, benefit from a characterization of what you were describing, exactly those models, from an information point of view? Which means, for example, oh, this disease is progressing in this way because I know there is an information loss that I can give a measure. So with the units, can be bits, but can be something different. But without, yeah. Because I see a lot of space there for a communication engineer to go there and say, oh, let's take these models and let's devise a measurement that can be, let's say, on top of these models.
Yes, that's for communication engineering. There's plenty of space. I could point those spaces,
I see that clearly.
But I love hardware, so I would focus better on the hardware and representing or...
I would say wetware.
Huh?
I would say wetware.
Yeah, wetware. Exactly. But, and I like to recreate those models. I like to grow those cells, which are all the time moving, and you try to catch them. So I like those computer game things, but introducing there the information theory and the loss of information will be highly beneficial. Yes.
I have a follow-up question kind of connected to what you just answered and what you just asked. Have you thought about including in silico models, like large-scale in silico models, as a transfer from your hardware to, for example, information theoretical modeling of what you're doing? So as Falco mentioned, we're doing large-scale human circulatory models. And I could definitely see this working to kind of transfer between your hardware and information theory, because like there's a lot we see a lot of benefit from those larger scale in silico models for example, CAR T cell treatment, where we've tried this out, that you can see different, like you can see how those parameters actually influence the movement through the human circulatory system. And for this in the endocrine system, I think it's really, it could be really interesting.
Yep, that's in silico models, they exist. There are groups which are separate work on them. Yeah, this is great to combine, but we are not so far yet, and will take some time, years. And maybe not for me, I don't like programming, so. Yeah, but in silico models, they are very useful.
Okay, thank you again for the nice talk.
Yeah.